Prize in Medicine was awarded to 3 scientists Monday for their work determining how the brain perceives places. The ceremony in Stockholm honored British-American John O'Keefe and Norwegian married couple May-Britt Moser and Edvard Moser. The three have worked on what's being described as the brain's inner. Who is currently based at University College London, has been studying that aspect of the brain, in particular place cells, since the 19th century.
Inside of your skull, know where it is, where it's going, and where it's been. This week, the Nobel Prize in Medicine was handed out to Professor John O'Keefe and Edvard and May-Britt Moser for their studies of the brain's internal navigation system.
The GPS system is made up of 4 types of cells: the place, grid, head directional, and boundary cells. The place, head direction, and boundary cells are found in the hypothalamus area of the brain, while grid cells are found in the entorhinal cortex, which is the main interface between the hippocampus and the neocortex. In the experiments where these cells were discovered, cell firing was measured. In terms of neurological cells, cell firing refers to the increase in electrochemical gradient of the axon. Place cells are neurons found in the brain which activate when we recognize a location we have experienced before. Different environments yield different firing patterns in the place cells, enabling us to differentiate between our environments. Place cells are also able to incorporate new movement information, which is known as path integration. This is important for keeping track of self-location during movement and is aided by grid cells. The sensory information is processed in various cortical structures before actually reaching the place cells, so the information received is a compilation of different stimuli. Being shown was the initial method that led to the discovery of place cells, conducted by measuring the cell fire rate emitted from a mouse through a tunnel. It was found from this that the cells fired in different directions in response to change in the available space in the environment. Boundary cells activate depending on the distance from the boundary and the direction from the individual. The optimum distance and direction varies between neurons. A boundary does not necessarily mean a wall, but could also mean a drop. Because of this, it is assumed that the cells are activated by sensory input and limit of movement. However, this is not being confirmed due to a lack of research. The relationship between the boundary cells and the rest of the GPS cells is currently unknown. The diagrams show that as the boundary approaches the cell's optimum distance, the fire rate is greater. It is also shown that the further the distance of the boundary cell's optimum from the individual, then it has a larger field of firing. Grid cells are neurons that fire when an individual traverses a small set of regions and become arranged in a periodic triangular array, as shown by the video. When grid cells lie next to each other, they usually form a grid-like shape and orientation, however will be distinguishable from other grid cells in a different part of the brain. Visual cues have a strong effect on grid cell alignments but are not solely responsible for their creation. Grid cells also appear on first recognition of a new environment, and as a result this is drawn upon when that environment is experienced again. Head directional cells are the neurons that fire according to the specific direction where the individual's head is pointed. It is frequently compared to a compass that doesn't require a magnetic field and doesn't necessarily point north, instead always pointing towards its optimum direction, which is predetermined within each individual cell. They do not fire according to eye movement, but instead the orientation of the head. It is currently unknown what causes the firing, as more research is needed. Conjunctive cells play a similar role to head directional cells and grid cells, but only when the head is directed towards a particular direction, like the head directional cells.
This is a sandwich, and we're going to hide it somewhere in this library, and our rat is going to attempt to find it. The first time he's going to have a bit of trouble, so he's not going to have any past experience of it. The second time, he should theoretically find it a lot easier. This is because he's formed connections between neurons in the place cells, head direction cells, boundary cells, and the grid cells. So you should find it a lot quicker. May-Britt Moser's and Edvard Moser's experiments, the rat was rewarded.
Alzheimer's is a neurodegenerative disease which has symptoms such as memory loss and an inability to recall recent events and places. The GPS cells help humans create a map of our surroundings, which provides space and time context in which new memories can be formed. The discoveries and understandings of these cells in the hippocampus and the entorhinal cortex are vital towards the research into Alzheimer's, as it is these structures which are first affected by the disease. By discovering these cells, we were able to better understand how Alzheimer's and other neurodegenerative disorders work, and therefore enable us to be closer in curing these diseases. Understanding the interactions between the different GPS neurons gives us greater understanding of spatial awareness and how we map out our environments. Being able to understand how these mechanisms enable us to work out how neurodegenerative diseases such as Alzheimer's affect spatial awareness. However, future research is still needed as areas of GPS are still not known as to how some parts work. For example, the relationship between boundary cells and place cells.
This week, the Nobel Prize in Medicine was handed out to Professor John O'Keefe and Edvard and May-Britt Moser for their studies of the brain's internal navigation system.